Hi everyone, my name is Juan Triana, and in this video I will talk about glycolysis, gluconeogenesis, and glycogen metabolism. Okay, first of all, let's talk about glycolysis. The glycolysis is the process by which glucose is broken down into molecules of pyruvate. This process occurs in the cytoplasm cell and is the first stage of both aerobic and anaerobic respiration. There are 10 steps involved in glycolysis, which can be divided into phases: the energy investing phase and the energy payoff phase. Okay, let's start with the energy investment phase that have the first four steps of the glycolysis. The first one is that the glucose is phosphorylated by the enzyme hexokinase to produce glucose 6-phosphate or G6P. The second one is that the G6P is converted into fructose 6-phosphate by the enzyme phosphoenolase isomerase. The F6P is phosphorylated by the enzyme fructokinase to produce fructose 1,6-biphosphate. The fourth is the FBP is split into two molecules of glyceraldehyde 3-phosphate by the enzyme aldose. Okay, let's continue with the energy payoff phase that have the last 5 steps of the glycolysis. The 5th step is that G3P is oxidized by the enzyme glyceraldehyde 3-phosphate dehydrogenase to produce 1,3-biphosphoglycerate. Okay, the sixth is that 1,3-BPG donates a phosphate group to ADP, producing ATP, and is converted into 3-phosphoglycerate by the enzyme phosphoglycerate kinase. The seventh is that 3-PG is converted into 2-phosphoglycerate by the enzyme phosphoglycerate mutase. The eighth is 2-PG is converted into phosphoenol pyruvate by the enzyme enolase. The ninth is that PEP donates a phosphate group to ADP, producing ATP, and is converted into pyruvate by the enzyme pyruvate kinase. And the last one, the 10th step, is that pyruvate can either be converted into acetyl-CoA to enter the citric acid cycle in the aerobic respiration, or be further metabolized in the fermentation pathway in the anaerobic respiration. Okay, so overall, glycolysis produces a net gain of 2 ATP molecules and 2 NADH molecules for each glucose molecule that is metabolized. Okay, now I will talk about the gluconeogenesis. The gluconeogenesis is a metabolic pathway in which the body produces glucose from non-carbohydrate sources, such as amino acids, lactate, and glycerol. This process occurs primarily in the liver and is an important part of the regulation of glucose homeostasis. Gluconeogenesis is essential for maintaining blood glucose levels during periods of low carbohydrate availability, such as fasting or exercise. The primary enzymes involved in gluconeogenesis are different from those involved in glycolysis. The process by which glucose is broken down to produce energy. This ensures that the two processes can be regulated separately, allowing the body to maintain a balance between glucose production and utilization. Okay, now the steps of gluconeogenesis can be summarized as follows. There are 5 steps. The first one is the conversion of pyruvate to oxaloacetate. In this step, the pyruvate produced during the glycolysis is converted to oxaloacetate through the action of the enzyme pyruvate carboxylase. The second step is the conversion of oxaloacetate to phosphoenolpyruvate. Here, the oxaloacetate is converted to phosphoenolpyruvate through the action of the enzyme PAPCK. The third step is the generation of glucose 6-phosphate. Here, the phosphoenolpyruvate is converted to glucose 6-phosphate through the action of the enzyme glucose 6-phosphatase. The fourth is the generation of glucose. The glucose 6-phosphate is converted to glucose through the action of the enzyme glucose 6-phosphate dehydrogenase. The last one, the fifth step, is the regulation of gluconeogenesis. Here, the gluconeogenesis is regulated through a number of different mechanisms, including change in the levels of hormones such as insulin and glucagon, and change in the levels of certain enzymes involved in the process. Okay, now as a conclusion, it is important to note that gluconeogenesis is not a simple reversal of glycolysis, as certain steps in the process are unique to gluconeogenesis and are not found in glycolysis. Additionally, some of the enzymes involved in gluconeogenesis have a different regulation than their counterparts in glycolysis, this allowing for the separate regulation of the two processes. Okay, to finish the video, I'm gonna talk you about the metabolism of glycogen. The metabolism of glycogen refers to the process by which glycogen is synthesized and degraded in the body. Let's start with the glycogen synthesis. The glycogen synthesis, also known as glycogenesis, is a process by which glucose molecules are linked together to form glycogen. This process occurs primarily in the liver and muscle cells and is stimulated by insulin. During the glycogenesis, the glucose is converted to glucose 6-phosphate by the enzyme hexokinase. The glucose 6-phosphate is then converted to glucose 1-phosphate and then to UPD glucose, which is used to form glycogen. The enzymes involved in this process of glycogen synthesis include the glycogen synthase, the UPD glucose pyrophosphorylase, and the glycogen branching enzyme. So, we have now the glycogen degradation, and this process also known as glycogenolysis. This is the process by which glycogen is broken down into glucose molecules. This process occurs in response of the hormone glucagon and occurs primarily in the liver. During the glycogenolysis, the glycogen is broken down into glucose 1-phosphate by the enzyme glycogen phosphorylase. This glucose 1-phosphate is then converted to glucose 6-phosphate, which can be used as an energy source. So, the balance between the glycogen synthesis and degradation is tightly regulated by hormones such as insulin and glucagon. The insulin stimulates glycogen synthesis and inhibits the glycogen degradation, while glucagon stimulates glycogen degradation and inhibits the glycogen synthesis. This allows the body to quickly respond to changes in glucose demand and maintain stable the blood glucose levels.